So today we will continue the discussion of the host side of the story, and that'll take us a little while still, because we're just beginning to scratch the surface here. The topic of today's lecture is host factors for disease severity and GWAS. GWAS, if you've never heard of this before, is an abbreviation that stands for Genome-Wide Association Studies. That was perhaps the first technique that ushered the era of high throughput genomic medicine and personalized medicine, and we'll learn a little bit about it today. The goal of today's lecture is to give you a lot of background in the beginning, and then lead up to the point where we can discuss this very short but interesting paper that, as you see, was published last week. That's one interesting thing about teaching this course, for me at least. It's basically, the topics are somewhat determined, or the articles somewhat drive, what I present, because interesting papers come up and sort of fit into these general categories that I want to cover. And this is definitely one that kind of bridges the gap between, a couple of fields. So genomic medicine, genome-wide association studies, a little bit of, statistics, and a little bit of human history. And this paper appeared in Nature, and the title of the paper, "The Major Genetic Risk Factor for Severe COVID-19 is Inherited from Neanderthals." The abstract is very short, so we can just read it. A recent genetic association study identified a gene cluster on chromosome 3 as a risk locus for respiratory failure upon SARS-CoV-2 infection. A new study comprising 3,199 hospitalized COVID-19 patients and controls finds that this is the major genetic risk factor for severe SARS-CoV-2 infection and hospitalization. This study is run through a multi-institutional international consortium called COVID-19 Host Genetics Initiative. Here we show that the risk factors conferred by genomic segment of about 50,000 bases that is inherited from Neanderthals and is carried by about 50% of people in South Asia and about 16% of people in Europe today. So, other than a catchy title, what this paper implies is that a large-scale study, genomic study, was able to identify a cluster of genes that are associated with an increased risk of developing severe form of COVID-19. There's another paper, and this is something that you guys are going to read for next time. This was an earlier paper that appeared in the New England Journal of Medicine, just a little bit more technical, where I'll just sort of skim what they're doing here. And this is one of the papers that is cited by the Neanderthal paper. So these guys performed a genome-wide association study involving just under 2,000 patients with COVID-19 and severe disease, defined here as respiratory failure, at 7 hospitals in the Italian and Spanish epicenters of the European epidemic. So they found, that the important part of the study, and you will sort of start seeing this as you look at these types of studies, is that there's always 2 sets of patients. If you're doing a association study, you need to have a set of patients that have the disease, in this case, severe COVID-19, and you need to have a matched, somehow, group of individuals that do not have this disease, so positives and negatives, so you can differentiate them. So they analyzed a very large number of single nucleotide polymorphisms, so just 8.5 million, and conducted a meta-analysis. And the typical report of this study would appear that they found a locus. So this all looks like gobbledygook if you've never seen this before, but RS 113.85.942 is a standardized way to refer to a SNP. I'll show you a database where you can look it up. It's located in chromosome 3, and another locus at chromosome 9, which were significant at the genome-wide level, with a slightly above 1 risk ratio. Interestingly enough, this paper claims that there's a locus that's associated with the ABO blood group. The Neanderthal paper actually says it's no longer the case. A larger study showed that this is not a real association, and but the other gene cluster stands, right? So we'll you will read this paper in greater detail, and we'll probably discuss a little bit about it next time. So now I'll move on to describe the necessary background. And if you recall what we did last time on the genomics, on the host genomics disease, on the host genomic side of the disease, was to discuss very simple, genetically speaking, Mendelian conditions, things like sickle cell, cystic fibrosis. And what was what sort of unified all those diseases is that their genotypic basis was very simple. And by simple, I mean it's a mutation in a single gene that leads to a well-characterized change in the phenotype, which then modifies something like an important function that leads to the disease. So, and the traditional approach for genotypic basis of disease was very successful for these types of for these types of conditions. So they were effectively idiosyncratic in the sense that there was no unified mechanism for finding them. That were discovered sort of serendipitously or through conjecture or just serendipity by individual doctors or researchers. That was done case by case. Typically, you already knew very well what the disease is. You knew how to diagnose it, so you could identify sick individuals definitively and quickly. And you would also have some understanding of genetics. So you would, for example, identify that a particular disease runs in the families. You might have some, simple analysis in a traditional technique. Traditional technique used there would be called QTL, or qualitative loci mapping. And then once you have all of this, you would eventually go and sequence the locus, find a mutation, and identify that it really is responsible for the presence of a particular disease. Now, we have a very different situation here because, for one thing, we don't have the time to do this for COVID, and we really also don't understand that much about the disease. At least we don't as much as we eventually will, or we do about other diseases. So there are a lot of anecdotes, a lot of different reports about specific conditions, but there's no unified mechanism that is agreed upon. What's severe COVID? What does it affect? So it's a kind of a diffuse condition. So modern approach that is more amenable to studying these complex situations where you have varied disease manifestations. You don't really have any idea a priori which genetic loci might be responsible for it. And you just want to use modern large-scale data mining techniques. You could call this reverse genetics. The traditional genetics would basically go from phenotype to genotype, right? Where you would, isolate everything down and do the genetics last. And here you sequence everybody and just perform a very large-scale statistical analysis. So ideally, you basically just obtain a large sample of individuals, positives and negatives. In this case, positives means having the disease, negatives meaning free of the disease. Disease will depend on the context. You will measure many possible variants. So at the extreme, you will just obtain the entire genomic sequence. That's usually not what people do. It's too expensive and unnecessary. So they will typically identify a large, but smaller than the entire genome set of variants of the sequence. It will be millions of individual variants. And they want to correlate that with disease phenotype or risk. So it's the opposite approach, sequence everything and then try to learn what might predict the disease. One might ask, how does this compare to the standard approaches? And two of the questions are, what are the benefits and what are the drawbacks? The benefits is that you can basically discover things. You're going into this without any specific hypothesis. So it doesn't, if your initial guess was wrong, you can still overcome this and learn whatever the data tell you. But that is also the drawback, because if you have no hypotheses and you have very, very large data sets or data sets that have millions of potential explanatory variables, a lot of the associations that you will end up finding might actually not have any bearing on the disease. So there's just a lot of noise and false discovery that could occur. We can correct for this, and people have developed very sophisticated techniques for doing so. And another drawback is that this is now effectively a computational exercise. So you need large computers, large sequencers, and sophisticated algorithms to do the analysis. So the complexity here is not necessarily the genetics of the biology, but rather sequencing and analysis. So let's, before we talk about COVID, let me sort of give you some of the basic intuition about how genotype might affect, a fairly diffuse phenotype. So for instance, one of the common, and I'm sure you've all heard about it at some point, that especially as you grow older, one of the markers for cardiovascular disease is increased cholesterol levels, and specifically low density lipids or LDL cholesterols. Our understanding of this evolves because, until recently, relatively recently, total cholesterols were measured, and that was taken as a value, as a single value to reflect whether or not you have some risk for cardiovascular disease. But then it was broken down into two components: high density lipids, which are good, and low density lipids, which are bad. So now, in addition to taking the total cholesterol, you also look at the relative composition of the two. And if you don't have enough high density and too much low density, that is bad. So low density lipids themselves are not abnormal. They exist in all people, right? It's a normal compound to find. And if you take a very large sample of the population, again, you could conceptualize it by thinking if you were able to measure that level in every single living individual, you'd have a very large collection of numbers, which you could then summarize by plotting a histogram, so the distribution of frequencies. And, it's a reasonable assumption that it'll look something like a bell-shaped curve or a Gaussian distribution, where you're going to have the smooth trend with a peak around where the majority of the values are. You're going to have a fairly rapid drop-off, and you'll have these values in the tails. So a couple of things you can learn by just thinking about what this figure tells you. One thing is that there's no such thing as a specific value that is normal, right? So, there's no one number that you can pick and say, this is the ideal level of LDL. You can assume that in most people, whatever is in the middle of this distribution, so maybe the range between 80 and 140 might be normal. Maybe it's narrow, or maybe you can refine it. But fundamentally, some range of values will be normal. And if you're within that range, you're probably okay. The interesting parts, occur as you start move away from the middle towards the, tail end of the distribution, right? So, for example, if you have very high low density LDLs are very low, that's probably abnormal. So now you're talking, abnormal means always relative to some distribution. And if you've ever looked at a blood test, for instance, you will see that the values that are reported are always, your value is 4, normal range is between 3 and 6, so you're good to go. You can smooth this histogram with a function, right, which idealizes, so this is just the continuous, normal distribution or bell curve that describes our model of how these low density lipid levels are distributed in the population. So this codifies the fact that we expect the large number of them to lie between, lie between 70 and 150, and so on the ranges. So now, forget about the histogram and look at this model. What can potentially contribute to the level of LDL or any other phenotype for an individual? Very broadly speaking, there'll be two categories. So you'll have the genetic component, and you'll have the environment. And it is very difficult to find any single phenotype. By phenotype, I mean, any measurement or condition that is exclusively determined by genes or exclusively determined by the environment. For the vast majority of them, the contribution from both of these components sums up to give you the total. So for instance, you can take it, genetically identical twins, which would presumably make their genetic contribution identical. And then you could have one that say, watches their diet and exercises, and the other that doesn't, right? That eats, Big Macs and drinks soda all the time. And obviously that would change, this measurement. And one of the ways to visualize this is that you can try to decompose the relative contribution of genes in the environment, right? So and this is what it might look like. So, and this is idealized. We can't measure this. So this is just, a concept or a model. So if we had the precise model, a precise understanding of how genes influence LDL levels or any other phenotype, this would be codified by the orange curve. One of the things you will notice is that the range of this curve is less than the range of the curve that is gene in the environment, which means that, and the way this is usually expressed in scientific literature is that genes only explain a proportion of the overall phenotypic variance. So variance is the spread of the distribution, say, 30 to 100, say, and the genes will only explain a portion of it, right? So for example, here, if you look at the overall distribution of LDL, maybe 70 to, like I said, 140 is a reasonable range. But if you look at the similar distribution of, that's given only by genes, it'll be, 80 to 130. So where do the others come from? And the others come from the environment, right? So if you have the environment effect only is also narrow, but if you add genes in the environment together, you get this contribution. The question is, would age be a factor that would change this distribution? For some phenotypes, it would, and that would actually be an environmental contribution, right? So yes, and you could also define, say, your LDL level dependent on the age of the subject. So you can imagine doing these types of analyses for, say, children, and then young adults, then adults, and then people of advanced age. A question is, this distribution considered for monozygotic twins, right? No, this is a distribution for all living people, right? There's no relation between them. This is just, if we were to measure everybody's LDL or a population's LDL, would see some distribution like this. And my question is, now that we've sort of have some definitions in place, last time I gave you a brief summary of a different paper from also a large-scale analysis, but this was not a genetic analysis. There was a large-scale clinical analysis where they looked at the researchers looked at which factors predicted severe COVID-19 outcomes, in this case, death. And some of the factors that they reported, if you recall, were age, sex, BMI, smoking, ethnicity, and IMD quintile. So you can already, just looking at those, you can see that there's already a mixture of both types of factors. For instance, and they're all mixed together. It's actually sometimes difficult to separate them. So, things like, sex is determined, biological sex is determined by genotype, right? It's not something that is strongly influenced by the environment. And age also, it doesn't really have anything to do with genetics. It's an environmental factor. Smoking is obviously an environmental factor. In this context, ethnicity is a genotypic contribution, even though it's confounded with other things. And then, if this was a measure of socioeconomic stress, again, it's obviously environmental. So even in this, short analysis that had no genetic data explicitly factored in, you already see that a mix of genotypic and environmental factors contribute to the risk of COVID-19 death. And in this case, age is the most profound one. And obviously that by itself is not a satisfactory explanation. It's just a proxy for something else, right? I mean, you can say that older people are at much higher risk, but then the next question is why? I mean, what is it that distinguishes old people in this context, in the context of COVID-19, from, say, young adults? And the answer to this question, as far as I know, does not fully exist. So when we talk about how genotype might influence phenotype, it is also useful to think in terms of distributions. And what you do now is you bring in genetic information. So in this case, we're measuring now some abstract phenotype. If you want to keep something in mind to contextualize it, just keep thinking of cholesterol, or then these will be reasonable values. And now you have 10 hypothetical individuals. You have their partial genomes, which is shown here. And there's one position in this genome that is different between these two sets of people, right? So person 1 through person 5 has a T at position number 4, and person 6 through person 10 has an A in this position. So now you're going to say, let's break them apart by what value they have, what nucleotide they have at this position, say a T or an A, and then let's plot the distribution of cholesterol for these two groups. And if you say something like this, where you see that individuals that have a T have an average of 100, and people that have an A instead measure an average 120, you might say, perhaps the presence of an A modifies this phenotype, increases it on average. Now, another important point to notice here is that we're talking about averages. So people with a person with a T can still have a high value of LDL. It's just less likely than it is for a person that has an A in this position. So when you say something like this, and this is of course a not very simple illustration because we're talking about a very small genome and idealized situations, you can say that having and assume that lower value is healthier, right? So you would interpret this as having an A variant would increase your risk for this particular disease, and you could say it would move the value by an average of 20. Now, on the other hand, if you have variation at a site where there's no movement in the phenotype, sorry, these averages get cut off, but they're identical, then you would say that there's no association between the presence of an A or a G at this position and having a change in your phenotype. Now, with these two pictures, you can already understand conceptually what a GWAS is. So GWAS is basically this, except there are thousands upon thousands of people. This table now is going to have millions of columns. And then you're doing these tests, seeing what happens to the phenotype for each individual column. So you do it on a computer in parallel, and then you do some sort of a, statistical procedure to identify which of the positions move the averages. However, and this is perhaps one of the key lessons I can teach you in this course, is that all of these analyses at best can find correlation. And it is tempting to say that if we found correlation, then that implies causation. So for instance, if I observe, in a large group of individuals, this relationship. So if I plot, say, average LDL levels, versus heart attack risk, and if I see that increasing LDL levels correspond to increasing heart attack risk, would I be justified in stating that increased LDL levels cause increased heart attack rate? So in other words, so this type of relationship between two variables is called covariation, right? So a change in one leads to a predictable change in the other, higher or lower. And this type of correlation is particularly simple kind. It's called linear correlation. So you can express them with a line. So, but can we say that X causes Y? And obviously not. So I'll give you a couple of silly counterexamples. So this is my personal favorite, where if you go through time, go back in the past, just say the 1700s and 1820, and estimate the number of pirates worldwide approximately. There were 35,000 in 1820, 45,000 in 1860, and so on. The number, the axes are a little strange because the number decreases over time. And then, on the y-axis, you're plotting the average global temperatures. Obviously, it would be silly and indefensible to claim that a decrease in the number of pirates caused an elevation in the global average temperature. But they're correlated, right? Obviously, there's no relationship between them, but they're correlated. So the question is, how do you establish causation if you have determined correlation prior? I'll get to it. It's there's a standard process where you basically, correlation is step one. So there's causation implies that there'll be some form of correlation. But correlation does not imply causation. So what you have to do is you have to actually, build the entire causal chain. I'll try to explain it. And there are also statistical inference methods which try to establish causation. So another one that was actually, you might think that I'm being specious about, presenting silly associations, but this is such a profound issue in science, and this is such a counterintuitive stance to take that even the New England Journal of Medicine, publishes papers that try to, drive home the practicing physicians and the readership in general that correlation does not cause causation. And they usually find, cute examples, and this one has to do with, identifying a correlation between national consumption of chocolate measured in kilograms per year per person, plotted versus the per capita or, Nobel laureates per 10 million population, right? And you can see that, at the far end of the spectrum, you have Switzerland, where there are a large number of Nobel laureates, and they also eat a lot of chocolate. And on the other hand, you have China, Japan, which, do neither, right? No Nobel laureates and no chocolate. And, here's the United States, United Kingdom, and so on. So you can, draw a regression line, say the slope of the regression line allows us to estimate that it would take about, 0.4 kilograms of chocolate, which is a pound per capita per year, to increase the number of Nobel laureates in a given country by 1. So for the United States, if we ate, 125,000 tons of chocolate per year more, we could expect an additional Nobel laureate. A less, silly example, which is actually something that is very common in medical inference, is, for instance, if you have this type of diagram where you have, just to lay down some more terminology, you have exposure, which means an environmental exposure in this case. And this is a very general term by basically, it could mean in this case, alcohol use or smoking. The outcome that they measure is a lung cancer. And then, for instance, you could, and confounder is something that that complicates your inference. So let me explain what this means. So for instance, if all you measure is alcohol use in the population related to lung cancer, you might find that increased alcohol use, leads or is associated with increased rates of lung cancer. However, if you don't think about this mechanistically, if you don't think, how could this happen, and you don't realize that alcohol use really shouldn't be directly influencing lungs, you might get liver cancer, but not necessarily lung cancer. What's happening here is that people that abuse alcohol perhaps have a concomitant rate increase in smoking. So people that tend to drink a lot also tend to smoke, and it is smoking that causes lung cancer. But if you don't this smoking is called a confounder, which means the actual relationship is, alcohol use has nothing to do with lung cancer. Smoking does, but alcohol use and smoking are related. And if you don't measure smoking, you don't measure the right variable, you will not be able to correctly infer that smoking is the root cause of lung cancer as opposed to alcohol use. And, in more general terms, what you want to do is you want to walk the entire path from gene to disease. And you want to take the straight path. You want to say a mutation in the gene causes disease. And for some diseases, that is true. We discussed some of those last time, the simple Mendelian diseases. But the traditional path is you go from gene, a change in gene will cause a genetic risk. Sometimes that implies disease risk, but not always. Disease risk sometimes, but not always, will cause the disease. And you can have confounders like the smoking. So you might have other genes that interact with the gene that you're looking at to modify genetic risk. These genes might also skip the genetic risk and modify the disease phenotype because they're not directly linked through it, but they might be linked through some interaction later on. Environment can also modify the genetic risk and the disease phenotype. And so this site is called gene-gene interactions, and this site is called gene-environment interaction. So it's a complex pattern. The reason I'm mentioning this is that it's very difficult. It's an arduous process to go from I found an association between something to I can explain what this means. So I can tell you, what the mechanism for a particular disease is. What does this mutation do? How does it affect the disease? So to show a causation, we must show a mechanism. So not just an association, but we must explain why. A set of mutations or a mutation will affect disease phenotype. One of the simplest, well, not simplest, but one sort of the gold standard techniques will be used in clinical trials, which we'll discuss separate when we talk about treatment. The entire purpose of setting up randomized clinical trials is to remove the effect of confounders. So you do it by design, so you can guarantee, statistically speaking, that there is nothing you have missed. And the way you do this usually is by randomizing over everything you cannot control, right? Because you don't know a priori what it is that may or may not influence the outcome. So what you do is you basically control for it by randomizing individuals and going to treatment or control. So we'll discuss this a little later. An empirically observed covariation is a necessary but not sufficient condition for causality. I'll give you just a few more examples of correlation not meaning causation. So the most obvious one is that there's nothing special about X and Y. If X is correlated with Y, you cannot say that X causes Y. So for instance, because windmills spin faster when there's wind, the correct explanation is that wind drives the windmills. But if you don't think about the mechanism, you can flip it around and say windmills cause the wind to blow. So this would be getting the direction of causation wrong. The unmeasured factor. So if C causes A and C also causes B, that does not mean that A causes B. And there's a great, there's an actual paper that made this a mistake. There's actually many more than one, but this is the one that I like to cite. So there was a paper that was published in Nature in the 1990s. And the claim of this paper was that young children who sleep with a light on are much more likely to develop myopia or nearsightedness in later life. So their conclusion was that sleeping with a light on causes myopia, which kind of makes you scratch your head because there's no obvious or even believable mechanism why this would, how this could occur. I mean, I suppose you can stretch it and say, if the light is bright, it stimulates your eyes at night and they don't rest and so on, but it really is a stretch. You sleep with your eyes closed. What does it matter if you have the light on or not? A later study that looked at this detail, actually presented a much more sensible explanation. So their explanation was that nearsightedness in children is very strongly predicted by whether or not their parents are nearsighted, right? So there's a strong genetic component to myopia. And incidentally, myopic parents, just because they actually, have a hard time seeing in the dark, they tend to leave the light on at night. So that was the connection. So what really happens in this case, A is you sleep at night with the light on, B means you develop myopia later in life, and C is both of these happen because your parents are nearsighted. You can also have correlation that goes both ways, right? So it's not, the number of lions in Kenya affects the number of gazelles in Kenya, because lions eat gazelles, but it's not something they can do indefinitely because, if they eat too many of them, the lions, that is, they will begin to die off. So you will have bidirectional causation. Increase in one will cause a decrease in the other, and eventually they stabilize. So it's not. Oh yeah. So let me look at the messages. That is correct. The FDA approval does not require mechanistic understanding. Typically, you need to have a mechanistic understanding to develop a successful drug. It's very difficult to just guess, but the FDA approval is based on clinical trials, which we'll discuss again, when we get to treatment, probably next time. Especially since it's been in the news, since President Trump got COVID last week and, received a lot of different treatments. So we'll discuss that next time, what the treatments are and how can you evaluate it. And of course, there's controversy. It seems to be everywhere these days. The Regeneron cocktail of monoclonals and so on. And finally, not to belabor the point, but there's a great actually website that all they do is they track ridiculous correlations, just to remind you that if you do enough tests, you will always find something by coincidence. So here's, a temporal correlation between per capita consumption of mozzarella cheese and the number of civil engineering doctorates awarded in the US. They seem to be correlated. US spending on science, space, and technology tracks the number of suicides by hanging, strangulation, and suffocation. The number of people who died by becoming tangled in their bed sheets is also pretty cleanly correlated with the total revenue generated by skiing facilities, and so on. So it is, and the reason I mention this is because coincidence is not something that you should easily discount. And coincidence especially becomes prominent when you're doing millions upon millions of comparisons. It's like saying, if I take a pair of dice and I roll them and I get 2 6s, that's reasonably rare, right? I don't expect it to do it many times in a row. But if you roll the dice, say 10 million times, getting a run of 5 6s is completely unremarkable. It's just the numbers game. So, and this is an example you should keep in mind when you think about genetic association studies, because there you're literally doing millions upon millions of such rolls. So each individual association might be unlikely, but if you start taking more and more of them into consideration, then some of them are likely to hit. Even the stop clock tells you the right time twice a day. Another component of genetic basis of disease is this concept of heritability. And heritability is primarily what distinguishes genetics from the environment. So phenotype is influenced by 2 major components, the genetic component and the environmental component. And heritability of the phenotype is an estimate of how much of the variability in phenotype and population is attributable to the variability in genotype. Importantly, the concept of heritability applies only to traits that differ between individuals. So if a trait exists in precisely the same form across individuals, all individuals, it may be inherited but cannot be heritable. So to give you an example, a trait that exists in the same form across all individuals is, say, the number of heads. We all have one head. Well, I suppose you can, find cases of conjoined twins, or something like that. but by and large, this is not a variable trait. So it doesn't make sense to talk about its heritability because it doesn't vary. And to estimate heritability, you do this very simple calculation. So the phenotype, which is P, this tilde means it's a function, probabilistic function of genotype in the environment. So G plus E just means they interact together. There's simple rules that you can apply when you look at random variables. So the mean, the mean of the sum is the sum of the means. So mean phenotype will equal to the mean genotype plus the mean environment effect. Variance is the second order one. So this is how far the distribution spreads. And the formula for the variance of a sum, the variance of the phenotype is the sum, the variance in genotype, so how much does it vary between individuals, plus how much does the environment contribute, plus this term, which is the covariance, which means how does genotype and environment influence each other. If you assume that genotype and environment don't influence each other at all, which is not unreasonable, I mean, that's sort of by definition, genotype, at least in this context, is something that occurs upon birth, and then or upon conception, and then you don't the environment doesn't influence it that much. Again, of course, it's not always true. So heritability is simply defined as the proportion of the total phenotypic variance that is explained by the genotypic variance. So it's the ratio. If there's no, and you can think about it this way, if there's no contribution from the environment, so all the terms drop out, environment is gone, there's no covariation, then heritability is 1. All the variability is phenotype is explained by genotype. If everything is explained by the environment, then G drops out, there's no variance G, there's no covariance, so H squared is now 0, right? So 0 means completely determined by the environment. 1 means completely determined by the genotype, and obviously the vast majority of phenotypes will be in between. So when you talk about heritability, when you hear heritability, that's what it means. That literally means the fraction. This goes back to the two curves that I showed you. It's basically, you had the white curve. So let's see if I can go back to it. It's this one here. So the variance in the phenotype is genes in the environment is basically the width of this curve. So how far can you go between the tails where there's still something left there. So here, the variability of genes in the environment maybe will be from 40 to 180, and here variability explained by the genes is only 70 to 150, right? So it's not 100%. So, and this is a good visual concept for variance explained. So how much of the total spread of the distribution by genes in the environment are explained by the genes. All right. And complex diseases are caused by a combination of genetic, environmental, and lifestyle factors, which is definitely true for COVID, most of which yet to be identified. And the vast majority of diseases that you're likely to experience in your lifetime fall into this category. They also include congenital defects, a number of adult onset diseases. So that would be things like Alzheimer's disease, asthma, Parkinson's, kidney diseases, osteoporosis, autoimmune diseases. And these complex diseases do not obey the standard Mendelian patterns of inheritance. So they don't have autosomal dominant recessive. It's much more complex. And that's because many, many different genes work together to determine the genotypic contribution. And the genotypic factors represent only a part of the risk associated with complex disease phenotypes. So sort of to break it down into categories, if you have Mendelian disease, most of its effect is determined by the genotype in the primary gene. You might have modification by other genes, severity of the disease, like cystic fibrosis. The environment plays very little role in this, and environment and genotype don't interact. Complex diseases have a very different picture. So typically you cannot narrow down the list of genetic factors to only a couple of genes. So generally low contribution to any single mutation. There's also a complex pattern of environmental influence. So it could range from low to major, and interaction can also range from low to major. So to give you examples of how the environment might modify the effect of a particular mutation, consider mutations in this melanocortin 1 receptor gene, MC1R, which is responsible for fair skin. Right? On its own, fair skin is a phenotype with no disease load associated with it. However, if you couple fair skin with an environment where there's a lot of UV light or sun exposure, these two combined will lead to the increased risk of skin cancer. Of course, exposing yourself to too much UV or sun will increase risk of skin cancer for everyone, with or without this mutation, but it might be enhanced by this mutation. So here, just to give you some examples of what this might be, the example that I personally can attest to is if you have this variant, a delta 32 deletion in one of the gene receptors, CCR5, on its own, it really has no effect. The only effect is manifested if you get HIV. And that only really mattered in the 1990s because, now everybody goes on treatment and it's much more benign, and as much as HIV can be benign. So carriers of the receptor deletion have lower rates of HIV infection disease progression. So this particular deletion was protective against HIV. Things like, if you have the slow metabolism SNPs in this, UGT1A6 gene, and you take aspirin, then it gives you increased benefit of prophylactic aspirin use, which is, if you know or if you don't know, it's taking aspirin is supposed to lower your heart attack risk, or ApoE, E4 allele, which coupled with cholesterol intake increases the enhances the uptake of cholesterol, right? So if you have this mutation, taking cholesterol intakes, it changes your cholesterol much faster, and so on. ADH1C, this version of alleles, if you drink alcohol, this enhances the factor of myocardial infarction, and so on. All right, this is good enough. So just keep in mind that it almost makes no sense to talk in very general terms about, how any particular disease is influenced by a genotype, because it's such a complex interaction. For most diseases, there's going to be a whole lot of individual genetic variants that modify the risk of this disease. There's the environment, and potentially interaction between the two. So you have to be very careful about how these studies are interpreted. Which brings me to a GWAS. So GWAS stands for Genome-Wide Association Studies. And what they do is kind of in the name. So they look at everything in the genome and they try to find associations, right? So it's in the name. They don't claim to do anything causal. It's just an association study. The distribution, and they focus on SNPs typically, so single nucleotide polymorphisms. The distribution of SNPs is determined in a large number of people, say hundreds to tens of thousands, with and without a particular disease. Then you measure millions of SNPs per individual, and then you test which SNPs tend to co-occur with disease symptoms. And if you find a degree of co-occurrence, then you make potentially a statistical inference, which is an educated guess, regarding the level of increased risk associated with each SNP. So I can give you, I can start off with a success story of GWAS, and this is perhaps the cleanest example of sort of personalized medicine that I can give. So there is this drug that most people will never see in their lives. It's called warfarin and Coumadin. It's a blood thinner. It's a prescription medicine used to treat blood clots and to lower the chance of blood clots forming. And it's typically used to treat, all kinds of heart conditions. So, atrial fibrillation, is given to people that receive artificial heart valves, people that have DVT, people that have pulmonary embolism. So anything that, wants to prevent blood clotting. Of course, if you thin the blood too much, then you have the opposite risk, of having internal bleeding or people dying from basically not being able to coagulate anything. And one of the things that was realized with this particular compound, with warfarin, is that different people responded differently to it based on their genotype. And it was a fairly simple genotype. So there was this New England Journal of Medicine study done in 2013, which is sort of a very traditional way, and this sort of brings in the idea of clinical trials. So they wanted to see if you could improve the performance of warfarin by deciding what dose to give to people based on which genes they have. So, typically, as you might imagine, you go, if you're prescribed a medication, you go to a pharmacy, you get the drug, the dose, the physician writes the dose, but it's never in common practice conditioned on what genotype you have. That might give you different doses based on your size, based on the severity of the disease, or based on, some other guidelines. But in this case, prescription is actually based on what genotype you have. So before you're given warfarin, you have to be genotyped to decide how much dosing to give you. So the study prospectively compared the effect of genotype-guided dosing with that of standard dosing on anticoagulation control in patients. And they did this randomized controlled trial involving patients with atrial fibrillation. So they had 455 patients. They divided them into two arms, randomly assigned to the genotype-guided group, and the remainder randomly assigned to the control group. So this randomness is what allows you to study the efficacy of the disease. And they found there were significantly fewer incidents of excessive anticoagulation, so adverse effects in the genotype-guided group. And they were also able to receive a better therapeutic range of the drug. And here's basically what a prescription might look like. And if you want to look at a record for this drug, there's an online database that contains information about these and other drugs that contains detailed information about prescription. So you have these two loci. So you have this CYP2C9, which is a microsatellite. So it's the number of repeats. 1, 1, 1, 2 is how many copies of it you have. It's not a SNP. And you have a SNP. And the dosage here in milligrams per day, right? So if you notice, you basically cross-tabulate this phenotype, GG, GA, and AA at this particular gene with, with different alleles. Now, these are not microsatellites, they're just different alleles. But you can see that the dosage orders differs by an order of magnitude. So if you have AA, monozygous at this genotype for A, and you have CYP2C933, the dosage is 2 milligrams per day, and here or 0.5. Here it's 5, right? So it's not a minor change. So that they improved. They were able to show that doing pharmacogenetic based dosing was associated with a high percentage of time in therapeutic range and lower adverse effects. So, what are these single nucleotide polymorphism variants that the GWASs look for? So these are point mutations. They're not just any change. They're point mutations which occur with sufficiently high frequency in the human genome. The most common form of genetic variation is a SNP. Others include copy number variation. In indels, population genetics tells us that most SNPs are neutral, which means they have no effect on any phenotype, but some are not. For instance, the ones that change the amino acid, non-synonymous, the ones that alter transcription efficiency, and so on. And these variations, this is something that will be important for the Neanderthal paper, is that a SNP sometimes doesn't describe just one change, but describes a whole cluster of linked changes in a particular region of the genome. Question is, one of the papers we'll have to read for today mentioned bypassing randomized trials. it is so critical for establishing its efficacy, why does the author want to bypass it? It's a matter of time and efficiency. It's a good and complex question, because it's always a trade-off in sort of emergency situations, which most people would agree COVID is. You have to make fundamentally a decision, which is, we don't want to call it a cost-benefit analysis, but I mean that's fundamentally what it boils down to. Because if for instance, deterministically, if you're guaranteed, if you know for a fact that if you do nothing, say 50 million people will die, and if you have the opportunity to lower this number by, say, 50%, then the question arises: is it worth the risk? And you have to be very, very precise about what that means. You have to be very, very precise about how to evaluate it. For instance, computer modeling is not an adequate way. It's not the only way to do it. It cannot be the only reason why we decide to do a particular intervention, say. And critically, it has to involve some idea of informed consent. An example that I can give you, for instance, that doesn't have to do anything with COVID, is the right to treat. That's what it's called, right? So, for instance, if you're a terminal patient, say cancer is the most common one, and you choose to take an experimental treatment, right? Because there is no other treatment available to you. That is also a difficult ethical question, but it is a decision that you can make. So it's, but it's in the context of COVID vaccines, bypassing randomized trials is a bad idea. And I think people have settled on this. And I don't think anybody's advocating seriously that, we rush an untested vaccine to market. So SNPs typically have two alleles. So they will have the wild type and the mutant variants, so commonly occurring base pair possibilities for SNP location. The frequency of a SNP is the frequency of the less common allele. So for instance, if the reference genome has an A and the minor allele is a G, so it occurs in 40% of the genomes, then the SNP will have a frequency of 40% or 0.4. So how much variation is there in healthy individuals that sort of represent the worldwide population and genetic diversity of humans? Until about 15 years ago, the answer would be we really don't know. One of the first very positive outcomes of being able to sequence genomes at a high rate was exactly the project to try to map what it is that exists in the human genomes in terms of variation. And the first iteration of it was called the 1,000 Genome Project. So it started in the late, like 2007, 2008, if I recall correctly. Right, so generated by several centers between 2008 and 2010. The paper that I'm discussing here was published in 2012. So obviously they've increased the size of this data set dramatically. So, we have millions of genomes now distributed across different centers. But this was the first paper that really laid down a reasonably clear picture of what it is that exists in terms of variation between people. So they generated 1,092 haplotype-resolved genomes, which means that the mutations were linked across the chromosome. So they combined different sources of data. They combined DNA arrays, so SNP arrays, which measured specific locations in the genome, and low and high density equality. Low and high density sequencing. So exome represents all the coding regions, so all the genes in the human genome. It is only about 3 to 4% of the total length of the genome, so you can afford to sequence it, with high coverage. And the rest of the genome in this particular study was done with low coverage for technical reasons, just because it was too expensive and too slow. not the case anymore. So once you have these raw data, you combine them and you map the variants. So you will have, these cartoon variants. So SNPs are just individual changes. So it's one nucleotide toggling to a different position. Indel is an insertion or deletion. So you have extra nucleotides or you're losing some nucleotides. And structural variant is something that involves a large reshuffling or rearrangement of the genome. So, gene being deleted, chromosome, pieces moving around, that kind of stuff. Statistical analysis to decide which ones are real and which ones are not. So here, statistical filtering. And finally, you basically produce this estimate of all the variants that exist in any particular genome. So for instance, there's 1, 2, 3, 4, 5, 6 genomes. They're diploid genomes. So, one chromosome and the other chromosome. And for example, in this individual, there's only one variant shown in this plot. It's an indel, and it only occurs on one chromosome, right? So that's a heterozygous variant. Second genome, you have, two heterozygous indels, and you have what looks like two homozygous SNPs. And the last genome, you have one heterozygous structural variant. This is probably the best summary of what they found. And this is basically a map of all the variants that they discovered in these 1,000 genomes. It's a fairly information dense plot, but it's, I think, worth understanding what it tells you. On the x-axis, you have the dimension, the genomic dimension of what's actually being measured. And this is a logarithmic scale. So as you move around, you start here in the middle of size 1. So SNP as a change of size 1. It's going to, the log of 1 is 0. So it puts you on the 0 scale. Anything moving to the right is green insertions. So you're adding genomic information. Anything moving to the left is deleting genomic information. And you have these bands that basically track the size. So, this is between 1 and 10 bases, between 10 and 100 bases, 100 bases and 1,000 bases, and all the way up to, 1 million bases, which are very, very large changes. So what you will see is, as you would expect, on the y-axis, you have proportion of variants. So how many variants were found? And again, this is log 10. So for instance, here they found, 70 million SNPs in just 1,000 genomes, right? So this already tells you, just dividing one by the other, that there is, 70,000 SNPs they found per individual, roughly speaking. And then as you go into larger and larger changes, you expect them to be more and more rare, because each of these changes is more dramatic than a SNP. Like adding 100,000 base pairs is going to be rare and dramatic, probably. And this purple line is proportion of variants that are novel. So novel to what was known in the previous literature, right? And they, basically, they've added a significant amount of information to all of the bands. Yeah, I'm going to skip this for the interest of time. Well, yeah, I will. All right. So you also have this broad dichotomy between SNPs that are common and rare. And that's exactly what they mean. It just, how common or rare they are in the individuals. So for instance, all of us have SNPs that nobody else has in the world, right? That's pretty good guess that at least a couple of SNPs are unique to us. They could be de novo mutations or, just some very, very rare variants. On the other hand, we also have a complement of common mutations, and a lot of it is determined by our pedigree, so by our ancestry, where we came from, or parents, grandparents, and so on. And those are old mutations that have risen to significant frequencies in the population. So rare SNPs are both rare and have high penetrance. So they have the potential to have a very, very significant effect. So early disease-causing SNPs were found using linkage studies from relatively small samples of related individuals. So basically, you're focusing in on a group of people that share the same genotype, or a significant proportion of the same genotype, to try to enhance your ability to find disease-causing mutations. So these approaches, however, were not successful in finding the genotypic basis of common diseases, so heart disease and cancer, which led to the establishment of this, perhaps the most prevalent explanation for what causes common diseases. And the current hypothesis is that common diseases are caused by common variants, or CDCV hypothesis. So common diseases, the hypothesis is that common diseases are caused by common variants. And implications, therefore, might be that individual mutations have low penetrance. So each individual mutation has a low probability of causing the disease. That's kind of obvious, because if you have a common variant that says occurs in the 40% of the population and it's linked to heart disease, a particular type of heart disease, it would mean that 40% of the population will have that heart disease. Now, we don't see that. We in fact see a very different manifestation of heart disease among even closely related individuals. So individual mutations are therefore very weakly influencing disease phenotypes, which means that you have to put together many mutations or many SNPs before they make any measurable effect. And this is an appropriate setting for GWAS studies if you have millions or, ideally very large sample sizes, to pick out these weak effects. So I already said this, all right. SNP with a 40% frequency, the population causes a highly deleterious amino acid substitution that directly leads to a disease phenotype, then 40% of the population would have this phenotype. All right, so said all of that. The alternative hypothesis is the CDRV, so multiple rare variations contributing to common disease rather than common variations. This has different implications. So if you have rare variants with strong effects, they're likely to be new because they, if they had been around long enough and if they have a deleterious effect, they would have been removed by natural selection. So rare variation is rare because they're being selected against due to their deleterious nature. Common variation are likely to be older and hence have been subjected to potential selective forces over time. Just because they managed to survive at an appreciable frequency, they're not as likely to have been subjected to negative selection. If you're interested, there are a number of websites that you can go to. So there's genome.gov, it's a US site, you have the UK site as well, that maintains a catalog of all peer-reviewed, sufficiently high-quality studies that have been successful in finding some association. So there's a nice picture of it. It's not, the most recent, but it's fairly recent. So what you see here is a cartoon representation of the individual human chromosomes, from 1, 22 on the X chromosome, and here's the Y chromosome. If you see a cluster of dots pointing to a particular location on this chromosome, that's an indication that there's some variants in this part of the chromosome that influence specific diseases, and color coding implies, tells you which diseases they are. So for instance, you can see the chromosome 6, for instance, despite being fairly short, especially compared to the others, has very, very dense clusters of associations with pretty much everything. And then chromosome Y, has next to nothing. It's difficult to study, for instance, and so on. So statistically speaking, how do we measure that a disease and a SNP are associated? We build what's called a contingency table, which is basically just a 2x2 table where you take all the individuals and you split them into 4 categories by 2 dimensions. If you measure in two dimensions, one is, are they healthy or are they diseased? And what disease means is very context specific. So we're looking at a specific disease. Say for COVID, that might be severe acute respiratory syndrome versus everybody else who may have had COVID but didn't develop the severe syndrome. So that's one dimension. The other dimension is a particular genomic location, and we'll want to measure whether or not they have a variant of this mutation or they have the wild type of this mutation. So healthy diseased, have SNP, do not have SNP. So H+ would be healthy with a SNP, D+ would be diseased with a SNP. H minus would be healthy without the SNP, D minus would be healthy, diseased without the SNP, right? And plus the total number of people with the SNP, and minus are those without the SNP. And we can define two quantities, because relative risk and odds ratio. And I'll give you numbers for those so you can see what those relative risk and odds are. So relative risk is very simple, right? So, you define the risk as, given that you have the SNP, how likely are you to develop the disease? So you look at all the people that have the SNP, there's 1,100 of them in this example. 100 of them have the disease, 1,000 of them did not, which translates to about 9.1% risk of getting the disease if you have the SNP. If you don't have the SNP, the picture is very different. Only 10 people out of 10,000 have this disease, which means that the risk is 1/10 of 1%. If you take the ratio of these two risks, you get relative risk. And what that literally means is if you have the SNP, you're 91 times more likely to develop the disease than if you don't have the SNP. That is a very, very strong effect. You almost never see this. This is, just to make the example very stark. You can also do odds ratios. So odds, if you've ever done sports betting or, cards or something like that, games of chance, what population does this data correspond to? This is just purely made up numbers. There's no population. It's just an example. And you should see this because the numbers are too round. You would never have a population where everything ends with a 0. Odds, the definition of odds are in the previous slide. So odds are basically disease. So the subtle difference is relative risk is the number of diseased individuals versus the total number of people that have the SNP, and odds ratios is a ratio of disease to healthy, right? So it's like, 3 and 2 odds, which means that, you have 3 chances out of 5 to win, so to speak. So odds ratios in this case, they're very close related to relative risk, so there's 100. Now, to give you an example where there's very little effect, now you have 100 out of 10,000 with a SNP get the disease, so the relative risk is, about 1%. Without the SNP, the relative risk is 0.7%. So it is lower, but it's just a little lower. This is actually a relative risk of 1.33 or odds ratios 1.3 would typically be a very significant finding for GWAS. So this is sort of what GWAS looks for, these really subtle effects. They're not looking for this because something like this is already known. So this is a Mendelian disease, right? This is something like, Down syndrome, even though it's not caused by SNP, it's a chromosomal rearrangement or, a polyploidy, aneuploidy, so an extra chromosome, or cystic fibrosis. This is more in line, with alleles affecting diabetes or something like this. And I really like this picture. It's from a relatively old review, but I think it summarizes, the 2 dimensions of, how common a disease is and how strong its effect is. So on the x-axis, you have, how common the mutations are, right? So here being, 1 in 1,000 individuals, that's very rare. And this is, half of the population, so it's very common. And it spans the range. And again, the scale is not linear, it's logarithmic. And then you have effect sizes, which you can think of this as odds ratios. So that's exactly what we talked about, right? So here it's a very small effect, and here it's deterministic, which means if you have the mutation, you're guaranteed to get the disease. And if you were to spread the sort of different diseases across this plot, you would see the following categories. You would have rare variants with small effects, which is empty. They exist, but we haven't, they're really difficult to find. You can have small effects for common variants, and this is where a lot of the common diseases like Crohn's disease, multiple sclerosis, diabetes, cancer risk factors would be. You have Mendelian diseases like cystic fibrosis, which would be rare, but very, very strong effects. And here you would have common variants with large effects, which is kind of uncommon, but one of the examples that people show here is ApoE4 for Alzheimer's disease. Right, so how do you find this common variation? um, uh, and this is basically, really neat high throughput sequencing innovation, because one of the one of the benefits of doing the 1,000 Genomes project was that we have a pretty good roadmap of which parts of the genome are very rarely different between populations and which ones are quite different. And it's very heterogeneous. Some parts of the genome are a lot more conserved than others. And if you're thinking about the best bang for the buck, you don't want to waste your efforts, looking at the part of the genome that is basically identical between most populations. You're not going to find anything there. You want to focus your resources at the parts of the genome that are diagnostic. So the HapMap project, went on to create a map of location and density of where the SNPs are. Then they sequenced very diverse, ethnically and genetically populations across the world to try to capture as much variation as possible. So in 2012, there were 11 different populations to basically try to identify variants that are unique to, say, European Americans versus Asians, versus African Americans, and so on. Of course, talking about North American study. Then you can have population-specific variation. And then what is important to understand in the context of the COVID study is that these SNPs usually basically come together as a group, right? So this is sort of like a package deal for a lot of these SNPs. If you have one, you're going to get, everything that's near it on the genome. And this is called linkage disequilibrium. If you've never seen this before, and I hope most of you have seen some version of it, one of the essential processes that generates genomic diversity and is a fundamental process in evolution is recombination. So recombination for diploid organisms means, so here's, if you look at the plot here, linkage with a family, you're looking at the chromosome of a child that, is a product of the chromosomes from, the parents. So they're the teal, the blue and the, whatever that color is, pinkish orange. There's one of the things that can happen is that through homologous recombination, the child will inherit a part of the chromosome from one parent, another part of the chromosome from the other parent. So you now have this recombinant chromosome, which means that if there were, whatever mutations existed, say on a maternal chromosome, are no longer together once you cross this recombination breakpoint. As you go through generations, this recombination has a cumulative effect. It is not undone. It is compounded by additional recombination. So as you go through, more and more generations, you will have shorter and shorter of shorter fragments of contiguous DNA inherited from one individual, right? So linkage between 2 points, and linkage literally means that this entire stretch of the chromosome was inherited from one individual. So all the mutations that exist in that individual exist in, let's say, the third generation progeny. And if you take this to the population level, first of all, you'll have an initial generation that's all mixed together, right? So they're all not starting together. And then as you go through more and more generations, you get more of a mosaic structure. So linkage disequilibrium, if you let this run for an infinite amount of time, then all the connection between all the mutations will be lost because they'll be broken up by recombination. So there'll be no history and no connection between any neighboring mutations. Linkage disequilibrium means that there still is a connection. So it's linkage. So linkage in this context means mutations that were inherited together and sort of passed on through multiple generations. And this linkage disequilibrium decays over time through recombination. And how quickly it happens, depends on population genetic parameters like population size, structure, number of generations. For instance, African populations tend to have lower linkage disequilibrium than European and Asian populations because European and Asian populations experience more recent bottlenecks. So sort of the amount of generations that they have to go back to find the most recent common ancestors fewer than in more ancient African populations. So there's specific, so this is an expectation of a complete equilibrium. if you have two alleles, one exists at frequency P and the other exists at frequency Q, then because you get to choose the genotype, diploid genotype independently, the frequency of having a homozygous small a is Q squared. The frequency of having a homozygous large a is P squared, and the remainder is the heterozygous. So this is what you would expect in population equilibrium. Any deviation from these relative frequencies tells you that it might be linked. So when we talk about linkage disequilibrium at the end of this lecture, that's what it means. Homozygous is represented by, so homozygous just means it's the same version, right? So two double small a's is homozygous, mutant, double capital A. Heterozygous is represented by 2pq, correct? Homozygous is P squared plus Q squared. So what you're basically doing, these two measures, R squared is the one that you'll see mentioned, is basically the frequencies of all these alleles that resolve to 0 if you're in complete equilibrium. So you can check as an exercise that if you plug in these numbers, P squared, Q squared, and 2pq in this expression, you're going to get 0 in the numerator. So genomic areas that have high linkage disequilibrium can be well described by a small number of tag SNPs. So instead of sequencing, say, 1,000 base pair, you only sequence one genotype, and then you interpret everything that comes with it as a group deal. So for instance, the HabMap project showed that over 80% of characterized SNPs can be captured with 500,000 SNPs. This is also the 23andMe approach. I don't know if any of you have done Ancestry.com or 23andMe, but they use chips, or at least that's the data that they have these SNPs. And as a byproduct of this, because of linkage disequilibrium, the outcome of a GWAS is an association, because you might have genotyped this entire stretch with one SNP. So you've genotyped this SNP, and it has no effect in the disease, but something that got carried along with it through shared inheritance is the actual unmeasured disease SNP. Yeah, we'll skip this. I'll also skip this. All right, so let's go back to the idea of statistical confounding. And this is, again, one of the most important points. And I really want to drive it home, because you need to know this pretty much for everything in life, because when you're given, a set of, facts or putative conclusions, the first question you should be asking is that, have they considered alternative explanations? So this is basically what covariate adjustment is. Is there an alternative explanation that is more plausible, more likely, or better supported by the data? And you can find all kinds of paradoxes, if you don't account for this, and I'll give you one or two in the next 10 minutes. So firstly of all, associations can be diluted or created by confounders. So age, gender, known clinical covariates. So there are known differences in phenotypes to ethnicities. I'll give you an example today. Allele frequencies are variable among populations. If you don't account for this, putting the two populations together will create false associations due to population stratification. And here's an example of what population stratification might look like. This is, again, a made-up example from a paper that I regret I failed to cite here. I'll correct it. So the question is, does a genotype contribute to disease? And it's hypothetical two populations, and you can do three different types of analyses. And, just to put this into context, think of population one, say, as, European, and population two, say, as, I don't know, East Asian, right? Just geographically different. And say we're measuring, the same two alleles, A and B, in both populations. And in this case, we have case control, disease, no disease, or the other way around. So here we have a situation when in population one, 90% of the cases have A, but also 90% of the cases have B. So there's no information. The same fraction of people that have the disease have A and B. So there's no association between the allele and the phenotype. The same thing is true in the second population, but goes the other way around, right? So the disease is, in this case, rare. So it's only 10% of the disease, right? So there's no association if you look at population 1 and if you look at population 2. But if you put them together, which you would think this is a good thing to do. You increase your sample size, you get more information, and simply by putting these two populations together, where there's no information, there's no association in either population. If you look at the numbers now, there's more, there's a higher rate of disease with A, apparently, than with B. So there's now this, association that pops out. So it shouldn't be there, but it appears because we now mix these two populations. Sort of a graphical way to visualize this is something that's called Simpson's paradox, which is a famous statistical paradox where you can, depending on how which part of the data you analyze, you can arrive at opposite conclusions. So say we're measuring some X versus Y, and we can do it separately in population 1. There'll be a positive association. As X increases, Y increases. In population 2, there's a positive association. As X increases, Y increases. If you put the two populations together, you'll now realize that the association is actually negative. As X increases, on average, Y decreases. This is, sort of a very, very clean visual example to keep in mind that, if you don't have a this has to do with representative sampling, right? If you don't sample, or I suppose, red or blue are fortuitous colors, since we're in election year. If you don't sample the global population and only sample, say, Republicans or Democrats, you may get an opposite conclusion of what the population is telling you. There was a question, what are your thoughts on genomic privacy? Do you think this is a significant barrier to analyzing genomic data? It's a very tricky question to answer. So genomic privacy is something that is only recently being considered as an important and an important issue in medical treatment, or privacy in general. And this is a very rapidly changing field because how much information is out there for everyone has exploded. Different people feel different levels of comfort about revealing their genomic data. And there's not sort of a standardized way across countries or even across states in the United States to deal with these situations. Generally, the United States is a pretty good genomic privacy policy. It's considered, personal health data, or PHI, personal health information. It's controlled by FISMA and HIPAA, which are two big federal statutes which prevent disclosure of your medical information without your informed consent. But there are caveats, because sometimes people can accidentally, well, not accidentally, but you can release information which you think preserves your genomic privacy, but in fact it doesn't. So it's a very, very tricky question. I think you should always be able to preserve the privacy if you want to. That's my personal view. And I have no interest in releasing my genomic information, without consent. I would feel very strongly about it. And I think, a lot of people would feel the same way. So let me give you another example that actually, puts confounding and Simpson's paradox in the context of treatment. So kidney stone treatment. So this is a paper from 1994, example of confounding, which gives you, again, a very simple example where there was a paper done even before that in the 80s that compared the relative rate of two procedures for treatment of kidney stones. So you have open surgery, which was trialed over 1972 to 1980. And in this study, that had a success rate of 78%. So 273 successes out of 350 individuals. The alternative treatment, the percutaneous nephrolithotomy, I'm not entirely sure what it is, but I would imagine it's a non-invasive treatment. had a success, or less invasive treatment, and a success rate which apparently looks higher, just on the surface of it. It's like 83%. If this is all the information you have, you would not be remiss to conclude that nephrolithotomy is better than open surgery. However, there is a confounder lurking, which is highly not obvious at first. So what if you take these two, this entire population of 700 cases, so 350 here and 350 there, and stratify them, meaning you divide them into two groups, basically, by how severe their condition is. So in this case, if the stone size is small, it's an easier case. If the stone size is larger, it's a harder case. And the boundary is 2 centimeters. If you look at this and just break the numbers down, 93% of cases of open surgery were successful, compared with just 83% of nephrolithotomy for small cases, for small stones, and 73% versus 69% for greater than 2%, right? So in both cases, open surgery, if you break it down, open surgery is more effective than nephrolithotomy. So how can it be? What's the confounder here? If for each one of those cases, small stones and large stones, one procedure is better, but if you put them together, the other procedure is better. What does that mean? All right, so how can open surgery be better in each of the two specific cases, but worse overall? And the confounding here is that the choice of the procedure is confounded by the complexity of the case. So treatment choice is not random. So basically, you preferentially will treat easier cases with nephrolithotomy, and harder cases with lower success rates are much more often handled by open surgery. So you're not performing a fair comparison, because the patients in the open surgery group are systematically more difficult cases. There's a whole encyclopedia of confounding in medical literature, which I might sort of borrow from a few more times. But the cases of how your intuition, an argument can be broken down by these kinds of more or less trivial complications, it's quite varied. And a lot of them, people and researchers make mistakes, not realizing that there's an issue with their analysis. So another example, which in the interest of time I might skip, but there's a paper that demonstrated as another exercise that lactose intolerance apparently can predict one's height. And it turned out, so I'll skip all of this, that in this case, everything was basically confounded by region. So, there's a geographic cline of height. So people say in Italy and Spain are on average shorter than the Nordic countries. And the prevalence of lactose intolerance is accidentally distributed across the same cline. Right? So they don't have anything to do with one another, but they have to do with the fact that these populations have just different compositions of alleles. There are other technical considerations that have to be taken into account. So you have to use statistical procedures to correct, for these millions of tests. We talked about p-values previously. So for for a typical paper, where a p-value is used, you will normally see cutoffs of say 0.05 or 0.01, or if you're being very, very strict, 0.001. And that roughly tells you how likely it is that if there was no association, or there was no result, you would have obtained, the same degree of significance. So it doesn't tell you how likely it is that your hypothesis is true, but it gives you some confidence, about the strength of statistical evidence. For genome-wide associations, because you're doing so many tests, one of the ways to correct for this, make this p-value tiny. So p-value for genome-wide association studies is something like 7.2 divided by 100 million. The question is, are there instances when the p-value is unreliable? Oh, yes. There are very, very many instances where it's unreliable. In fact, p-values have sort of, fallen out of favor with the statistical crowd, not because there's something inherently wrong with them, but because it's very easy for people to misuse them. Like with anything else, right? If you use the tool correctly, you will get a desirable result. But with p-values, because there's so much confusion about what they mean and what they don't mean, a lot of people inadvertently make mistakes. So the other type of inference, in statistics, there is the sort of two major approaches to doing inference. So there's something called the frequentist approach, and there's something called the Bayesian approach. I don't know if I might need some slides for this, but Bayesian approaches basically so a frequentist approach, it's kind of easy to conceptualize, and it sort of goes back to like basic natural science. So you want to measure, you go to the lab and you want to measure, some relationship between, say, force and acceleration. I'd say so, right? So you do this measurement, you establish some hypothesis, and then you repeat this measurement, right? You repeat it many times and see how often do you get the same result. And it's this idea of replication, right? So if you're able to run the same experiment over and over again, how often would you see the same result if there was no association? With Bayesian statistics, it's very different. It's this idea that you have prior beliefs about what should and should not be true. This is your prior expectations. And then as you look at the data, it will modify your prior beliefs. It will make things more or less likely, right? So, in a typical assumption, if you have no information, then everything is equally likely. Things is more or less associated, 50-50. If you have some other information, you can incorporate it, and then it can be modified by the data analysis. So for Bayesian statistics, you will typically have something called, posterior predicted values or posterior probabilities. It is somewhat harder to interpret in some sense, but in many cases, you can sort of achieve the same ends with different approaches. All right, so let us now get to the study that I opened the lecture with. So the major genetic risk factors for severe COVID-19 inherited from Neanderthals. So as a background, dramatic differences in COVID disease progression. You have the gamut from asymptomatic to severe respiratory distress. Major risks are age, being male, and some comorbidities. We saw that before in other studies, but those factors do not explain all the variation. So what about host genetics? Since the beginning of the epidemic, large consortia have been set up to study the genetic basis of COVID from the host side. And there's one particular study that this paper uses called COVID-19 AG. So AG stands for humangenetic.org. I'll present the result, and then we can go look at this at the website. So this was actually taken from the COVID AG studies. So this type of plot, and well, it's called a Manhattan plot because it kind of looks like the skyline. This is a very, very dense, high-level representation of a genome-wide association study of a very, very large analysis. So it's large in two dimensions. First, it studied nearly 1 million individuals. So they had a group of 3,200 hospitalized COVID-19 patients, and they compared it to about 900,000 population controls. One of the reasons we're able to do these analyses is because US, UK, and a few other places have set up or basically large-scale, biobanks where you prospectively collect data. You can send people in. They can agree to deposit their data into these repositories. For example, the largest collection is called the UK Biobank. It's about 500,000 people that's routinely used in these types of studies. And it was specifically designed to enable exploratory analyses for diseases and conditions that we don't know much about, including COVID. So this was already in place for population controls. Obviously, the 3,200 hospitalized COVID-19 patients were new. But this type of analysis builds off an extensive infrastructure that was put in place to study exactly these types of associations. So that's a great example of forward thinking that allows accelerated discovery when it's necessary. On the x-axis, you see highly compressed representation of the human genome. So this is chromosomes. They're different sizes, so that's the size of them. And then what you see here is just this blur of dots. So each dot, they overlap, there are millions of them here, represents one SNP that was tested. So there was something like 8 million SNPs that they tested. So they've compared, performed 8 million statistical tests, each with a specific p-value. This dotted line here, or dashed line, represents the minimum significance. So the largest p-value you can allow, or rather the smallest p-value that will allow you to interpret results as significant. So this axis goes backwards. So on near 2 means that the p-value is 0.01. 4 means, because it's a log 10 scale. This is 1 in 10,000. This is 1 in a million, 1 in 10 million, and so on. Anything that rises above this line has a statistical muster. It's a pretty striking pattern because you see that there is a large number. It's actually hard to tell how many dots, but there's possibly 100 dots that are all in the same spot here, this locus on chromosome 3. They're all together, they're all above this level, and there's nothing else, right? In this analysis, no other site or no other chromosome has crossed the threshold of significance. So this is highly unusual because in a typical disease association study, you will kind of have a smattering of dots, from all over the chromosomes that go over this level of significance. And here I'm just going to quickly jump off the presentation and go over to COVID-19 HG. I'll reshare my screen in a moment here. All right, have it up. So here's it's actually a very detailed, information-rich website. You can request access to information. You can do your own analysis if you're a researcher. But let's go to the results here real quick. And that they have updated. So this was released in September 2020, and you can see that they've updated the analyses, and you will now recognize these types of Manhattan plots. Don't worry about this. This is a significance level plot. But here you see the same cluster of points at level 3, and a few others show up. And these are the cohorts that were used here. They have done different analyses. So this was hospitalized COVID versus non-hospitalized COVID. This is a much more typical, type of plot that you might see with, just a few dots scattered across the map. This is hospitalized COVID versus population, and so on. Let me switch back to the lecture. So this is the Nature paper is only 2 pages. It really has 3 figures. So and a lot of it is just a simple interpretation of what other people have done. But what the authors of this particular paper noticed, which is hard not to notice, is the spatial clustering of all these variants. So they all come together. They're all next to each other in the chromosome. So what does it mean? So the first thing they wanted to see is, are these variants in linkage disequilibrium? Right. So in other words, do they tend to be inherited together? So what's shown here is a zoomed in version. We zoom in on just chromosome 3. So this is the genomic megabases, coordinates in chromosome 3. So we're looking at about 1 million bases shown here. This is the linkage disequilibrium scale. So 0 means they're not linked, so they're mixed up by recombination, and 1 means they're perfectly linked. So they always occur together. So if an individual has 1, it's going to have a whole lot of other variants. So let's read the caption. So this is linkage disequilibrium between the index risk variant. So this allele, this SNP called RS3544562, a genetic variants in the 1000 Genomes Project. So as you see, this is the gift that keeps on giving. So the Genome Project allows you to associate variants in standard populations. So red mark genetic variants are where the alleles are correlated to the risk variant, and the risk allele matches this Neanderthal genome. So in addition to human genomes, we've also sequenced several ancestral genomes, including a fully assembled several Neanderthal genomes. So what they observed in this study is not only do all of these variants come together in individuals that have one variant, but they're also shared with the Neanderthal genome almost identically. So this black bar is an almost complete match to this section of the Neanderthal genome, which is about 150,000 years old. But you can think about it, this entire cluster of variants is very, very old. So it's hundreds of thousands of years old. And it was basically passed without change as a group, from generation to generation, and now, having this particular cluster of mutations seems to predispose individuals to poor COVID outcomes. So under the x-axis, so if you see these types of plots, these are individual genes, right? So this is the genomic structures. So you have 1, 2, 3, 4, 5, 6, 7, 8, 9 genes. The way these are plotted, if you have a tall bar, this is an exon, so this is genetic coding. If you have just a line, this is an intron. So if you kind of eyeball it, most of these variations fall into what looks like the exon regions and maybe the introns of this LZTFL1. So I'll conclude. I don't want to switch screens too much. So, after I'm done presenting this paper, we'll go back and I'll show you where to find, what this RS is and where you can find what some of these genes do. So what this paper noticed is that the genetic variants which are most associated with severe COVID-19, they all exist in chromosome 3. They live in this region, between coordinates 45,859,651, 45,909,002. AG19 is the version of the human genome, according to which this mapping was done. They're all in high linkage disequilibrium, so they're all inherited together, very strong. There's about 50,000 base pair long region. Some such long haplotypes, so when they all inherited together in linkage, this is called haplotype. The question is this also the chromosome also involved in HIV immunity? Yes, that's very good. I'm glad somebody noticed. So yes, all of these are receptors in immune cells. So some of these things, it actually might be interesting, and I'm sure people have already done it, to dig in and see what that means. But yes, it's definitely something, that reminds me of HIV. So good catch. The genetic variants, so such long haplotypes have entered the human population by gene flow from Neanderthals or Denisovans that are extinct hominins that contribute genetic variation to the ancestors of present-day humans some 40 to 60,000 years ago. So they noticed this linkage. They noticed that it shared its ancestry, and almost all of the variants were identical to the Neanderthal genome. So they wanted to look deeper into it. So here's a cartoon from Wikipedia. So take it with a grain of salt. I don't have any reason to believe there's anything wrong with it, but this is a very, sort of coarse depiction of the long range evolutionary history of humans and their ancestors. So, the modern day ancestors, depicted here is orange, this large population. So, the most recent common ancestors of us and related species was in Africa 2 million years ago. Then there was, this split, Homo heidelbergensis, heidelbergensis. I apologize for mangling the term. My Latin isn't very strong today. Which then split into Neanderthal and Denisovan. You can see the migration. You can see the Denisova cave, where Denisovans get their name, which then went on to East Asia, what is now Australia. And so what we're seeing here is, Neanderthals. So we had a separate lineage, but Neanderthals contributed some genomic information to modern humans, right? In fact, if you if you do something like 23andMe or Ancestry, one of them, or actually I think both of them, will report how many variants you have from the Neanderthal genome. It'll sort of place you in the relative rank where you are in the human population. I did 23andMe. I think for me, it's, I have something like, I'm in the 60th percentile. So, not terribly remarkable as far as Neanderthal ancestry goes. This paper asks the question that the stretch of variants that seems to be conferring susceptibility to COVID-19 is shared with the Neanderthals, but where did it come from? Did it come from Neanderthals, or did it come from the most recent common ancestor of Neanderthals and Denisovans? They make a simple argument based on recombination rates and estimated date to say that it's unlikely to come from a common ancestor. So they infer that the variant came from the Neanderthals. And then what they do is build a simple tree. This is a phylogenetic tree. I think the last time we saw them was maybe lecture 1 or 2 when we're looking at the origin of bats. The origin of coronaviruses and the evolutionary history in bats. But this evolutionary tree takes a short fragment of the human genome, so that something like 50,000 base pair. Each of these Roman numerals represents one of the common variants in the human population. So it's a haplotype, right? It goes 1, 2, 3, 4, all the way to 21. So there's at least 40 of them, 50, or 40 something. So these are just different common variants that are spread throughout the human population. These variants 1, 2, 3, and 4 are the ones that carry the susceptibility variant. So they're the ones that have the mutations that seem to be associated with severe COVID. And these guys, the Altai, Chagriskaya, and Vindija, are the lineages of several different Neanderthal genomes. So in this particular part of the human genome, what we see in modern humans today that seem to be susceptible to severe COVID is most closely related to Neanderthals. So this is another way to confirm the origin of this particular group of variants. And then the last thing they present is the geographic distribution of where those haplotypes are. So see, they're almost absent in Africa, very rare in South America, low frequency in North America, relatively low frequency in Europe, although there's some variation. Somewhat prevalent, but not too frequent in the Middle East. But then if you look at East Asia, so India, looks like Pakistan, maybe Vietnam, maybe Bangladesh, they're now almost up to 50%. But in a neighboring region in say mainland China or in Japan or in South Korea, they're almost not detected at all. And then you go to Australia, Papua New Guinea, they don't have anything for Indonesia, but that is also quite prevalent. So this is immediately an interesting story because this factor, if there is a significant geographic disparity about how these variants are distributed, then you might, think, for instance, what if, if there's an excess of severe cases in India compared to neighboring countries? Might some of it be due to the fact that you have more susceptible populations as opposed to, more or less effective controls or better treatment? And that's exactly one possibility that the authors suggest. And they take it a little further. I would like to commend these authors on their restraint about speculating, what the results mean. They were actually quite reserved, which is rare and should be commended. Neanderthal risk haplotype occurs at a frequency of 30% in South Asia. Sorry, I misspoke. What did I call? I said it was. Sorry about that. Let me try. Let me share my screen again. I confused West and East. That's embarrassing. There we go. Let me reshare my screen. Right, so sorry, this is, West Asia, not East Asia. It's almost absent in East Asia. And there's some evidence that other Neanderthal fragments have been affected by selection in the past. At the moment, we can only speculate about the reason for this, one possibility being protection against other pathogens. So you make a little bit of a reach, you might say that perhaps, in East Asia, this haplotype is decreased in frequency due to negative selection because it made you more susceptible to coronaviruses, other coronaviruses, which, if you recall, are quite ancient. So have probably been infecting the human population for as long as humans existed, in one form or another. So perhaps that's what happened. There's no evidence for this, but it's a possible hypothesis we could explore. It is also currently not known which of those variants actually causes the disease, or what it does. If it does, why does it do so? And to quote this paper, with respect to the current pandemic, it is clear that gene flow from Neanderthals has tragic consequences. All right, so I'll throw up the paper that I would like you to read for next time, which is it's just a very technical but well executed genome-wide association study for COVID-19, which gives you more technical details, so you can kind of get a little bit more in the woods and see the types of analyses that people do. And then we can begin next lecture with a brief discussion of this paper. In the meantime, what I'm going to do, I'm going to switch. I'm just going to spend 2 more minutes showing you where you can get information on the individual SNPs. So if you can go, if you want to learn what some of these SNPs do, so NCBI, which is the Central Repository of Biomedical Information run by the United States government, has a database that is specifically geared towards identifying SNPs. So you can type in the SNP that was referenced in this paper, any other SNP, they're all going to have standardized notation. You can search for it, and you will find some pretty basic information about what this is. So that's a single nucleotide variant. It's a change from an A to a G. It's located on chromosome 3. It's annotated as being an intronic variant, upstream transcript variant, and the frequency in different populations is quite varied. So, 2% in the Vietnamese population, 45% in this population, which I'm not immediately able to tell you what it is. There's a gene that's associated with LZTFL1. You can click on another viewer. It's going to load up the genomic map for this gene, and you're going to have all the known variants. So there's, and you can click on them and see what, it's going to take you, structural variants. Now, the next thing you can do is you can go, if you're curious about what some of these genes do, you can go to NCBI Entree, which is sort of the omnibus search for, it searches all the databases, and you can type in LZTFL1. I just spell it right. Let's see what it tells us it is. So there we go. So there's the information for this gene. So it's a leucine zipper transcription factor like 1. You can click on this and learn, where this gene is. This gene encodes a ubiquitously expressed protein that localizes to the cytoplasm, blah, blah, blah. Now you can do this, you can dig in deeper and look at the literature and various clinical information. So there, for example, there are clinical information. So ClinVar are the variants of clinical significance that have been mapped to this genome. And here's going to be a summary of studies where you have individual variants, where they occur, does it change the protein or not? Was it associated with any particular type of syndrome or disease? What effect it has, likely benign, uncertain significance, and so on. Was it reviewed? Has it been properly annotated? So let's see if there's COVID here. Nope. There's no COVID in this table yet. And then similarly, you can look for other genes. So you can see what's, for instance, CXCR6 does, which is the next gene there. So this is a chemokine receptor. See, they're part of a signaling pathway that regulates T lymphocyte migration to various peripheral tissues and promotes cell-cell interaction with dendritic cells and fibroblastic reticular cells. So now you might start seeing some, light bulbs going off in your mind because, we spend a lot of time talking about T cells and immune responses and how important it is to have a robust and effective T cell response for disease clearance. So one potential hypothesis would be, is it just because you have this cluster of mutation that sits in the middle of genes that are associated with T cell signaling? The obvious hypothesis to explore would be, do these mutations change something about the efficacy or the rate or the durability or the breadth of the immune response? The question is, there's a question in the audience, how do you know exactly the coronavirus susceptibility in different patient populations is really variant-based? Could just be dependent on compound health complications.
I realized I kind of asked the question before I finished typing. So what I meant to say was that how do you know that the region located on chromosome 3 is not just the result of the particular effect of COVID on the particular health complications that patients have? How do you know it's not just based on the health complications?
That so what typically you will do in these types of studies, if they're done well, and we'll discuss it next time based on this New England Journal of Medicine study, is that you definitely want to account for other confounders, right? So for instance, so you have to make sure that you cannot explain it away with anything else. One possibility would be, so for instance, if all the individuals that were sick also happen to be old, and they also happen to be enriched in this chromosome 3 by accident. But in a study like this, because this is a very common correction, they will allow, they'll make statistical modifications to ensure that this contribution of chromosome 3 is independent of anything else. So it's in addition to in addition to anything else that might be complicating it. It's in addition to, say, diabetes or obesity or advanced age, or all the other things that we already know matter.
I see. Thank you.
Sure. All right. That's all I got for today. Thank you for great questions. And I will see you in a week.
Thank you, Professor.
Thank you.
Take care.
Thank you.